Hello, this is Lisa Seidman, and this is the first in a series of 6 videos that are about the calculations involved in biological solution preparation. Videos 1 and 2 introduce some important terminology and concepts. Video 3 talks about the calculations where concentration is expressed as a simple ratio. Video 4 gets into calculations where concentration is expressed as a percent, and Video 5 talks about calculations when concentration is expressed in terms of molarity or millimolarity. The final video, Video 6, talks about calculations for multi-component solutions. So let's get going with Video 1. So in this video I'm going to introduce 2 very important words. Those are concentration and amount. And we'll get into those in more depth in later videos. But before I get into these 2 kind of lab words, let's talk about a setting that might be a little bit more familiar, like a blueberry farm. So let's suppose that we have a blueberry farm. Here's the blueberry bushes arranged in rows. Let's suppose that the farmer for this farm knows that he or she gets roughly 100 pounds of blueberries every year from each row. So per 1 row. Okay, that's great. Now suppose the farmer wants to know how many pounds of blueberries to expect from the entire farm. Well, to figure that out, the farmer needs to know how many rows there are. So to keep this simple, let's suppose there are 10 rows. So if there are 10 rows, how many pounds of blueberries will the person get? Well, that's a pretty easy calculation. If you get 100 pounds of blueberries, we'll just say here, blueberries, in 1 row, per 1 row, then we just multiply by 10 rows. And you know from algebra that the rows will cancel, and you get 1,000 pounds of blueberries. Okay, I think that's a pretty easy concept and a pretty easy calculation. So now let's go to a more lab setting. And let's talk about amount and concentration. Amount is just like we think about it in normal English. So for example, 100 pounds of blueberries is an amount of blueberries. Or we might have, say, 1 dozen dz of eggs. That's an amount. To make it sound more like a lab, let's suppose we have 10 grams sodium chloride. That's an amount. Sodium chloride is ordinary table salt. Maybe we have 1 milligram KCl. By the way, this was, I believe, grams there. Okay, all of these are amounts. Concentration is different. Concentration is a ratio. It is an amount per a certain volume. So in the numerator, we have amount, and in the denominator, we have a certain volume. Now, as an aside, a chemist might sometimes have mass or weight here in the denominator, but that's less common for us as biotechnologists. So we're going to just talk about volume in the denominator. And for us as biologists, that volume is almost always going to be a volume of water, or something derived from water, like a buffer, which is a solution that keeps the pH constant. So we say it's going to be something aqueous, where aqueous is of water. So for example, if we go back to our 10 grams of sodium chloride and dissolve that so that there is, say, 1 liter of water in 1 liter of water, that would be a concentration. So this is a concentration. Now note I did not write of water. A lot of times that people might in a shorthand skip saying water because water is assumed much of the time in a biology setting. So this is a concentration. 1 milligram of KCl per, let's say, 1 mil, again, would be a concentration. Okay, I just want to make a comment now. about the blueberry example. So before we were talking about blueberries. So we said there were 100 pounds of blueberries per 1 row. Now, of course, that's not a concentration as we defined it here, because we said that a concentration is an amount per volume, and this is not a volume. But the kind of calculations we did in the blueberry example are very much like some calculations that we're going to be doing over and over and over again when we talk about concentration. And since it's very likely that you understood the blueberry example, it's very likely that you're going to understand the calculations we do in the lab. Okay, so let's think about this a little bit more. Here we have 2 tubes, and let's suppose in this tube we have 10 mils of water, and here we have 5 mils of water. And in this water there are some stars. Now let's suppose the stars represent, I don't know, grams, milligrams, micrograms. For right now we don't care what they are. So we have 10 stars. You count them, there are 10 stars per or in 10 mils. Here we have 5 stars. If you count them, there are 5 stars in 5 mils. Observe, the amount of stars in the 2 tubes is not the same. However, the concentrations are the same, because we can divide 10 stars, 10 by 10, 10 divided by 10, the numerator and the denominator divide by 10, and we get 1 star per 1 mil. And here, if we divide the numerator and the denominator by 5, we get, again, 1 star per 1 mil. So the amount of stars in the 2 tubes is different, but the concentration of stars in the 2 tubes is equivalent. All right, so let's go to another example. Okay, here we have 2 more tubes. This one has, again, 10 mils of volume. And this one has 5 mils of volume. If you count the stars, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 stars in 10 mils. This has 10 stars also, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this has 10 stars in 5 mils. And in this case, even though the amounts of stars are the same, concentrations are different. Because this again is 1 star per 1 mil. And this, if we divide both the top and the bottom by 5, is 2 stars in 1 mil. We can go to yet another example. And in this case, the volumes are the same. So we have again 10 mils here, 10 mils here. But since the amount of stars is different in the two, the concentrations will be different. So this is going to again be 1 star per mil. This is going to be 5 stars in 10 mils, which is the same thing. We divide top and bottom by 5. 1 star per 2 mils. So these two are not equal. All right, now to make sure that you've got this, that you're really understanding this, here's a little problem to solve. Which of the following have the same concentration of sodium chloride? A, B, C, or D? It could be more than one. A has 1 mg of sodium chloride per 2 mils, 30 milligrams sodium chloride per 60 mils, and so on. So what I'm going to do is I'm going to wait about 4 seconds, and I would suggest, if you want to, you could pause this video and you could try to figure this out. Which of the following have the same concentration of sodium chloride? And I'm going to pause, so I'm going to stop for about 4 seconds to give you time to pause the video. And then in about 4 seconds, I'm going to go through the answer to this and discuss it briefly. So stop the video if you would like to solve the problem. Okay, so let's take a look at this. This one is 1A. Let's start out with A. It's 1 meg of sodium chloride for 2 mils. All right, that's pretty simple, straightforward. Now let's look at B. If we look at B, we could divide the numerator and the denominator both by, say, 30, which would give us 1 milligram sodium chloride in 2 mils. So that is the same as the first one. Let's look at C. If we simplify it by dividing the numerator and the denominator by 100, we get 1 milligram sodium chloride. But now we have per 3 mils. So this one is not the same. A and B were the same, but C is different. Let's look at D. Well, this is a little less obvious. If we take both the numerator and the denominator and divide by 780, we can again get 1 milligram in the numerator. And it turns out if we divide 1560 by 780, again we get 2 mils. So A, B, and D are the same concentration, but C is different. Okay, so the next video we'll move into some different ways to express concentration, because this is a way to express concentration as we've done in this video, like it is here, but this is not the only way to express concentration. So see you in the next video.